Alright, so this is video 2 of the series on photosynthesis. So we talked about the process of photosynthesis and what's happening and being able to follow the elements or the atoms through this process. And we've talked about water and carbon dioxide, light, glucose, and oxygen. But we added a couple of molecules here, NADPH and ATP. And so all I want to do is point out that it's a little bit more complicated than as simple as knowing this equation. And you need to have a deeper understanding of what's going on here because it's going to come into play in later decisions you're going to be making. So I want to talk very quickly about how do you know if the rate of photosynthesis is increasing. And so there are a couple things that we can measure, and some of them are what we call direct measurements. And some that are, or really one that is indirect. So the two direct measurements that we can make are the oxygen. So we can measure the oxygen produced as a function of this photosynthetic rate or photosynthetic process, or we can measure CO2 consumed. But I will say that this becomes a little bit more problematic in our understanding when we consider that plants are also doing cellular respiration. I'm just going to abbreviate CR. Are also doing cellular respiration. So in addition to producing oxygen, they're also consuming oxygen. And in addition to consuming carbon dioxide, they're also producing carbon dioxide. But the way that you need to think about this is that they can't do the photosynthesis at high levels unless there's a light source. So if we're talking about plants that go through the normal typical day-night cycle that we have here in Virginia, there are going to be times when the photosynthetic rate is reduced, and therefore, but cellular respiration, which is not dependent on light at all, aside from the production of some of the things that are needed for it, those rates are going to stay pretty consistent throughout through time, whereas the rate of photosynthesis is going to increase and decrease as a function of the amount of light available to it. Okay, so that's something to think about when you're in your designs. But the one indirect method, and I'll label it 3. The indirect method is actually to measure the biomass. So measure the biomass, the mass of the living parts, and sometimes this is called dry biomass. And really what you're measuring is the glucose that's made. Because the oxygen is released and it's gas, so it's glucose that we're measuring. And so the glucose that is produced, I'm going to do this in a different color here. The glucose that's produced can be used in a variety of different ways. So one is that it can be used immediately for cellular respiration for the plant, so that it's going to be making adenosine triphosphates, or the energy molecule for itself. It could use it for heat, so as a byproduct of cellular respiration. Not all of that energy is converted to ATP, some of it's for heat, and that's really important for enzymatic reactions to prevent this process called denaturation, which you may want to look up. That's also in chapter 2. It is probably something you talked about in 9th grade bio as well. But you might want to refresh by just flipping to enzymes in chapter 2. Chapter 3. I think it's 3.6. And lastly, which is the most important for us in this case, is building polymers. Building polymers. And these are the polymers. Poly means many. So these are the large molecules made of lots of subunits, which happen to be called monomers, that we can use. So the polymers that we're talking about here would be things that you're familiar with, called starch or cellulose. So starch and cellulose are made by the plants from the monomer glucose. So glucose is a small molecule, C6H12O6, relatively small, and starch and cellulose are both made of thousands of these glucose molecules that get stuck together. And they get stuck together through a process called condensation reactions or dehydration synthesis reactions. And if you don't know what those are, make sure you add those to your need to know and see if you can look into it or ask me for some help. We'll talk a little bit more about that. So that's really what's important, because these are the polymers or the glucose itself that we're going to consume. And if we're trying to grow food, that's quite important. So I want to talk a little bit more about temperature, though, because temperature is really important to control here. This is one of our controlled variables. This is something that can change, but we want to try to maintain it, or at least keep record of how the temperature is changing. So I'm going to ask, this is my abbreviation for temperature, how does temperature affect the rate of photosynthesis? And I'm not going to answer that here, because I know that I could go into a lot of detail, and this is something that you could very easily look up in your book in the section about photosynthesis. But questions that you should think about is, how do we control for this? How do we control, or maybe a better word here is maintain, the temperature of the environment? And in our classroom, it may be variable. In a greenhouse outside, it's also going to be variable. So how do we maintain that? How do we hold a certain temperature without adding a whole lot of cost to it? And really, how do we optimize it? And this may depend on the species that you pick. How do we optimize the temperature? So you need to know what the optimal temperature is for your plants. Another question that comes up is how does light intensity affect the rate of photosynthesis? Light intensity. And really there's another factor with light. It's not just light intensity, but it also has to do with the spectrum of light, or the different wavelengths of light. So how does the absorption spectrum come into play here? Also, how does CO2 concentration affect the rate? I'm going to abbreviate photosynthesis as PS. How does carbon dioxide concentration affect the rate of photosynthesis? And, you know, we talk about carbon dioxide, we're talking about light. The other input is water. So are we concerned with the amount of water? Maybe even temperature of water. There are lots of other qualities of water that are really important. And yes, you might be thinking, well, we're in an aquaponic system. The entire medium is water. But in what ways and where can we lose water? So where is the water loss, and how do we accommodate that? And how do we prevent water loss? I think those are all great questions to think about. And as a follow-up, you know, how does water get into roots, and how does it travel? How does it travel up a plant? All right, so those are things that you probably should be able to answer now, based on what we did in class. And do plants, or maybe let's say do different kinds of plants, different species of plants, have adaptations to different environments? And it probably seems quite obvious that yes, of course they do. Nowhere on Earth is it the same. It's not the same everywhere on Earth. So, but we have plants in a lot of these places. So, what's different about them, right? So, things to think about or to look into is, and I expect you to be able to do this, is looking at adaptations of how plants, how they get water, because they might have different ways of getting water. They have different chemical pathways or different modifications of photosynthesis themselves. They're different pigmentations. So we talked about chlorophyll, but not all plants have the same levels of chlorophyll, and that's why they have different colors. If you go out into the woods, and you often do, like on your way to 7-Eleven from school between classes, you might notice that there's an underbrush of plants that's near the ground, and then you've got an empty space of where there's not really a lot of greenery for a good 10, 20, maybe more feet, and then you've got the tops of your trees. But there's a different quality of light in the underbrush than there's on the top of these plants in the canopy of the woods there. So the plants have different pigmentations. They're picking up different wavelengths of light. They're reflecting different wavelengths of light. And even in a single tree, the leaves at the surface actually are worse at absorbing light and even reflecting light. Instead, they allow light to pass through their leaves even better, so that their own leaves lower down can still absorb some of the light, and it's not totally reflected away from them. And the last question I want to bring to you, but really is meant to get you to be thinking a little bit more, is what else? What else might be carried in the water? What else might be carried in the water? Okay, so for example, I'll give you one. I'll tell you one thing. There's something called chelated iron that travels in the water. It can dissolve very easily into water. Think about the properties of water we talked about. It dissolves into that water and then can follow the water into the plants, and that iron is really, really important. So the plants use that iron as part of their chlorophyll pigmentation to absorb light, to kick off the entire process of photosynthesis. And without it, the plants don't look green. They don't look green because they're not reflecting green light, because they don't have the iron in their chloroplasts, in their chlorophyll, to absorb the other wavelengths of light that they need. And the plants start to actually look yellow. And when we eat those plants that are high in iron, or that are darker green, because they have more chlorophyll, like spinach or any dark greens, that's a good source of iron for us, which is actually used in our hemoglobin. Heme means iron, so our red blood cells have lots and lots of iron in them, which attract and hold on to oxygen. They have the high affinity for oxygen, which moves oxygen around our bodies through our circulatory system. So it's really quite important to understand what else can be traveling through the water and what we might need to add to our system to ensure healthy plants, which then would lead to healthy humans. And that's the whole point of this project, right? To provide fresh, healthy foods to people. And so it really comes down to some of these, what we call trace elements in the system. Okay? And I'll leave you there. I think that's enough questions, probably lots of things to think about. And I'd point you to your book. You got some resources for some of these questions. Use the internet for others. But look in the chapter on photosynthesis, which is in chapter 3. And there's some discussion on some of these. Others you might need to look up, and those that you still can't find answers to, make sure you ask in class. Okay?